Hi, everybody. We are having some technical problems, so Rachel signed out. She's going to sign back in. So give us just a second.
Okay, my computer audio, webcam. Can you hear me?
Yes, I can hear you just fine.
Start my webcam. Okay.
Now let's see if it gives you.
Okay. Yeah, can you maybe?
Hang on just a second. Give it just a minute and it should come on.
Okay, show my screen. Okay.
Yes, yay!
Okay, so should I then do full view now?
Yes, go ahead and do full
see,
view.
like me, because right now I don't see me or anything, so.
I see you, I see me, and I see your screen.
Okay.
So we should be good. If we get just 1 minute, give me just 1 minute. Thanks everybody for being patient. Sometimes we hit some technical glitches here, so I'm hoping you all can hear us. If you give me somebody would just put in the questions, yes, we can hear you. We will get started. And I will get off camera and let Rachel take it away. Rachel has been studying mangroves down in this area. She's up in New York now, and she's working with some folks up there on some of the projects she had started down here. So Rachel, you want to take it away?
Yes. So again, you can see my screen and you can everyone can see and hear me.
I can see you fine and I can hear you, and we're being recorded.
Okay, perfect. So thank you for the wonderful introduction, and thank you to all who are joining in. I'm excited to talk about the research that I did during my master's, which was investigating regional scale differences in the physical structure of mangroves at an expanding range limit in northern Florida. So I wanted to start off by talking about species tropicalization, which is a global movement of tropical species towards poleward latitudes in response to primarily warming air and sea temperatures that is caused by climate change. And the authors here depicted tropicalization out here on this map. And with species tropicalization, we're going to get an increase of tropical species at temperate regions, which is indicated by the red arrow. However, with tropicalization, we're then also going to get a decrease of temperate species, with tropical species moving towards poleward latitudes, which was indicated here by the blue arrow. And with species tropicalization, this is not only going to have harmful impacts on the surrounding environment, but also on humans and potentially our economy. Now, a notable example of species tropicalization is happening with mangroves. And mangroves are a group of tree or shrubs found along coastal intertidal areas throughout the tropics, subtropics, and now in temperate regions. And mangroves provide many unique ecosystem services, such as regulating, cultural, supporting, and provisioning. So for example, for regulating, mangroves provide coastal or shoreline protection from storm damage and hurricanes. For cultural, it is estimated that mangroves bring in a multi-million-dollar industry across the globe for ecotourism, as well as for supporting, mangroves support a nursery and breeding ground for many fish communities, as well as other inverts and fish and bird species. And lastly, for provisioning, mangroves are the reason people may have a roof over their head or a meal on their next plate seen across the globe. Now, when we talk about mangroves across the globe, the latitude extent of them is pretty large, typically ranging from about 25 degrees north to about 25 degrees south, indicated here by the blue arrows. And within this range, this is where we have both the highest number of mangroves as well as the highest species of diversity. And it has been well understood and documented in the literature that air temperature is one of the main primary determinants of mangroves' latitude distribution. However, it is a common misconception that it is just air temperature alone. It is also due to the reasons such as the decrease in the intensity and frequency of winter freeze events. And because of this, mangroves have now been expanding to poleward latitudes previously outside their range limits, indicated here by the pink arrows. Now, I know I talked about mangroves on a global scale. However, for the focus of this talk, we'll be focusing on mangroves throughout the Gulf of Mexico. And mangroves have been present in the Gulf of Mexico for over 100 years. However, due to those periodic freeze events, this has limited mangrove expansion. And in 1989, there was a major freeze event in Texas that caused a mass die-off of mangroves. However, since then, our winters have been generally more mild, which has facilitated the expansion of mangroves throughout 4 major hotspots throughout the Gulf. You can see here throughout Texas, Louisiana, into Mississippi, and both northern coasts of Florida, especially for the Avicennia germinans or the black mangrove. And it is anticipated that in 2010 in Florida, another major winter freeze event occurred. However, since then, winters have been more mild and has facilitated the expansion for the Rhizophora mangle or the red mangroves. More specifically, for this talk, I'll be focusing on this northern hotspot in Florida, which was the focus of my study site. And what's really important about my study site is we have both species of mangroves, unlike the rest of the Gulf of Mexico, where red mangroves aren't as abundant. And lastly, although it's well understood about global scales that drive mangrove presence absence, there still is a lot of uncertainty about what climate factors affect the abundance or distribution of mangroves on a small or micro scale. Now, about almost 3 decades ago, the northernmost transition between mangroves and salt marsh was documented in the literature to be Cedar Key, which you can see here right on the map in Florida, and mangroves south of Cedar Key throughout South Florida, where environmental conditions are favorable, we get mangroves that can look like this. And on average, mangroves in South Florida are about 9 meters tall. And here's an actual picture that I took in one of my field sites in Miami. And I personally have seen mangroves as tall as 15 meters. However, when you get to the northernmost range limit of mangroves in Florida, specifically in Apalachicola Bay, we get mangroves that look like this. They're shorter, wider, and shrubbier. And in a recent publication, mangroves in northern Florida average about 1.3 meters tall. Now, mangroves have been present in northern Florida since the early as 1940s, and we have both documented herbarium records as well as aerial imagery that show mangroves have been present as early as 1941. You can see here out on Dog Island, as well as 1950, and throughout the years. And I wanted to show one of the actual documented herbarium records. Here is the Avicennia germinans, or the black mangrove, that was collected in 1950 on the northern side of Dog Island. Now, I'm not the first person to be studying mangroves in this region. However, it is still relatively a very young region for mangrove research. And a very important paper that was recently published was by Snyder and others in 2021, where her and her team set out to document the presence of all the mangroves in this region. You can see up here, starting in Alligator Point, going all throughout the barrier islands in Apalachicola Bay and up into St. Joseph's Bay. And you can see here that the dots on the map indicate the presence of a mangrove. The red dots indicate the presence of a red mangrove, and the black dots indicate the presence of black mangroves. And although this was a really large and amazing effort, this still leads to some knowledge gaps about what is the population structure of mangroves in this region, which leads me to my research objectives. So I wanted to take it a step further from Snyder's paper and now characterize the physical structure of mangroves across this region. I hypothesized that I would expect taller and higher stem densities will be found in my southernmost site and decrease from going to low to high latitude. And one thing I just want to make note is when I'm talking about latitude in my region, it is across a 1/2 degree of latitude. So really looking at microscale differences in the physical structure of mangroves. I then also wanted to determine if differences in the regional nutrient content explain variations in the physical structure. Specifically, I wanted to see if there were differences in the foliar or leaf nutrient content using leaf nutrient stoichiometry as a proxy comparing total nitrogen and total phosphorus across my sites and between species. And lastly, as I mentioned earlier, air temperature is a primary determinant of mangroves' latitude distribution. So I wanted to see if there were differences in the regional freezing temperatures that explain the physical structure of mangroves. So first, starting off with the mangrove physical structure. So now this map might look very similar, but now this is a field map of my study sites, which span about 90 kilometers from Apalachicola Bay in the west, and then going north to Oyster Bay in the east. And we have my northernmost site of Piney Island, then going south to Porter Island. And if you're familiar with the area, that's not too far from Panacea. Going down to Alligator Harbor, then my intermediate site of Dog Island, and then Goose Island and Unit 4 located out on St. George Island, and then lastly, my southernmost site of Pilot's Cove. Now, before I get into my field methods, I wanted to take everyone on a field trip and show you some pictures of the mangroves at each of these sites. So starting off with my northernmost site of Piney Island, this is characterized by one lone red mangrove, or our mangle, across this entire island. Then going south to Porter Island, this site has Avicennia germinans, or the black mangrove, and this site is characterized by only 2 black mangroves. Then going south to my site of Alligator Harbor, similar to Piney Island, this site only has red mangroves. And mangroves across this field site, we have mangroves scattered across different mudflat and oyster reefs. My intermediate site of Dog Island. Now this site has both species of mangroves. We have both red and black mangroves. And just want to give a big thank you to Jen Bueno for the wonderful aerial imagery. And one thing that we are seeing with mangroves at their northernmost range limits is that they're like growing in clusters or different patches, which you can see here I've highlighted in the red circle. But what you see up in the air is a bit different once you actually get down on the ground, because there are some areas on the islands where it looks like just a sea of mangroves. However, there still are some other areas on the islands surrounding these clusters of mangroves is salt marsh vegetation. And my next site is Goose Island. This is located out on St. George Island. This site, similar to the others, only has red mangroves. And this site is where I think you can mostly see that distinct patches of mangroves highlighted here in the red circles. But similar to before, what you see up in the air is different once you get down on the ground, because surrounding these patches, you get these hundreds of seedlings and saplings of mangroves, which can make some field surveys a bit more difficult. Right next to Goose Island is Unit 4. This is located right near the St. George Island Bridge. And similar to before, this site also only has red mangroves. And last but not least, my southernmost site of Pilot's Cove. Like Dog Island, this site also has both species of mangroves, and this is one of my favorite sites. It is also my largest site, because what you see up in the air is what you see down on the ground. This is where we have the most mangroves, the tallest ones that I've been seeing. But there's also areas on the islands where you have these distinct patches just surrounded by the salt marsh vegetation. So how did I actually go ahead and measure the physical structure of mangroves? So I measured the heights of both species of mangroves, and they were measured for 2 different class sizes. So for all the mangroves taller than 1 meter, they were classified as tall, and for all the mangroves shorter than 1 meter, they were classified as the short class. And they were all measured in within a 10-meter diameter plot, as well as with a 1 meter squared quadrat. So I would go up to the patches of mangroves at my site, set up my 10-meter diameter plot, and within that plot, I measured every single mangrove that was taller than 1 meter. And then for all the mangroves that were shorter than a meter, they were measured within the 5 quadrats. And now you may ask, why didn't I just measure all the short mangroves in the plot? Because as you can see on the picture on the right, there are some areas on the islands where it would be very easy. However, you do get to some areas where it is just nearly impossible for me to have measured every single short mangrove. It would mean measuring thousands of stems, hence why I had to use that quadrat method. So now looking at the results. I know this may seem like a lot, but I'm gonna walk everyone through it. So this is just looking at now the tall class size heights of mangroves. And we're not seeing too much differences, but in general, we're seeing that mangroves, the tall mangroves are at about 150 centimeters. So you can see here on the bottom axis, we have my sites. So starting with my southernmost site of Pilot's Cove, going up to my northernmost site of Piney Island, and then broken up between species. Very similar for now, looking at the short class of mangroves, we see a bit more variation. And now looking at stem densities, or how many of those mangroves were now within those plots. So similar to before, again, we just have my sites, my southernmost site of Pilot's Cove, going up towards Piney Island, and now looking at the stem densities, or how many mangroves, and we see a lot more variation. And just one thing I'd like to note is just the letters indicate significant differences. And the most dramatic differences that we are seeing is with the short class. And one thing I would like to know is just the change in axes on this graph is going almost up to 500. So in other words, I counted almost 500 short mangroves within the 1 by 1 meter quadrat. And although we are generally seeing the red mangroves grow taller than the black mangroves in this region, we are seeing that black mangroves are dominating in the stem densities within the plots. And this possibly could be due to differences in growth rates or differences in establishment times. So next, looking at nutrient content. In other words, I wanted to see if differences in nutrient availability had some sort of an effect on the physical structure of the mangroves. So to do this, I looked at the foliar or leaf nutrient composition, and this was compared across sites. And for the focus of this talk, I'll just be talking about total nitrogen and total phosphorus. So to do this, I collected green mangrove leaves out in the field, took them back to the lab. They were freeze-dried, homogenized, and then they went through a series of elemental analysis for total nitrogen and also total carbon. And then samples were prepared and digested using a discrete analyzer for total phosphorus. So now when looking at between species, so regardless of site, so all the red mangrove leaves that I sampled compared to all the black mangrove leaves I sampled, there were no significant differences. And again, just looking at total nitrogen to total phosphorus, they're multiple ratios. However, when we then break it up now between sites, so this first graph here is looking at all the red mangroves I sampled across sites. Similar to my other graphs, Pilot's Cove, my southernmost site, going up to one of my northernmost site of Alligator Harbor. Again, letters just indicate significant differences. And the only significant differences that I'm seeing for the red mangroves is between my 2 sites of Unit 4 and Dog Island. And the reason for this is, if you're familiar with the area, you may know that Unit 4 is located out on St. George Island. It is located really close to people's homes, so it's really close to urbanization, runoff, and other pollutants, compared to my most secluded site of Dog Island. And then when we look at now just all the black mangroves that I sampled at each of the sites, Pilot's Cove, Dog Island, and then Panacea, there were no significant differences. And lastly, trying to tie it back to my original question is, does differences in nutrient content explain the physical structure of mangroves? So for example, the height. So now when we look at total nitrogen to total phosphorus against height, I did measure the heights of the mangroves that I did clip the leaves from, and there were no significant differences. So overall, in our region, there is no indication as of now that nutrient differences among my sites and between species is what's driving the physical structure of mangroves. So that brings me to my last research question, is looking at freezing air temperatures. And before I get into my field methods, I just want to give a little bit background of what we have in the literature right now. And first, just looking at the black mangroves, we start seeing leaf damage during freeze events as low as -4 degrees Celsius. And then we see near mortality of black mangroves when temperatures drop below -0.7 degrees Celsius. However, for the red mangroves, to my knowledge, there are no field-based observations that have been published. There have been some controlled greenhouse and laboratory experiments. However, there is no field-based freeze damage and mortality thresholds in the literature for red mangroves. They are not as abundant throughout the Gulf of Mexico, so this does make it a bit more difficult compared to the black mangroves. So now for my methods, I use iButton air temperature loggers that were deployed across my sites to investigate micro-scale differences of winter air temperatures. So you can see here as my loggers on the left, they were deployed on the field for about 3 months. And then once they came back to the lab, they were all offset corrected to a known calibrated logger. You can see here in the center that was set to the National Institute of Standards and Technology. Out in the field, all loggers were deployed 1 meter above the soil surface in both mangroves and salt marsh vegetation, and then also at the fringe, so right at the water's edge, and then also in the interior sections. You can see here on the picture on the left, here's one of my iButton loggers hanging in a mangrove. Very similar in the middle picture, again, 1 meter above the soil surface. And then for the loggers deployed in the salt marsh vegetation, they were on PVC poles. And all loggers were deployed to measure air temperature for every 20 minutes. And all loggers were deployed to start recording on December 15th of 2022. However, if you live in Florida, you may know that just 9 days after I deployed these air temperature loggers, the National Weather Service declared a winter freeze event across the United States known as Winter Storm Elliott. And for my field site specifically, from December 24th to December 26th, minimum temperatures dropped below -4 degrees Celsius. And although that there was a major winter freeze event on Christmas Day in Florida, this did not stop surfing Santas and snowmen throughout Florida. So now looking at the data for my air temperature loggers. This first graph is just showing the data taken from December 24th. So this was the coldest day of the freeze event. And then on the bottom axis, we just have latitude. So again, starting at my southernmost site of Pilot's Cove, going all the way up to my northernmost site of Piney Island, and against minimum temperature. So this was the lowest temperature that my loggers recorded on December 24th. And there were no significant differences between latitude and minimum temperature. But I also do only have about 12 or 15 points on this graph. However, when you break it up now looking at between vegetation and then fringe versus interior, so location of where the loggers were placed, we actually do see a significant difference against mangrove fringe and salt marsh fringe compared to the salt marsh interior sections. So the fringe loggers were actually warmer compared to the salt marsh interior. And taking it a little bit step further, so now when we look across the 3 coldest days, so December 24th to December 26th, there actually were significant differences, and the fringe locations were significantly warmer by 1.25 degrees Celsius than the interior sections. And this possibly could be due to just closer to the tidal waters, providing a bit more warmth. And then also when looking at mangroves were warmer by 0.3 degrees Celsius than the surrounding salt marsh vegetation. And I know this might not seem like a really high number. However, these microscale temperature differences are really important because it's just showing how microscale temperature differences are playing a role in the distribution of mangroves across the barrier islands in this region. So because I did not anticipate for there to be a winter freeze event, this then provided an opportunity to conduct a post-freeze burn and mortality assessment. So similar to how I measured the physical structure, I again set up the 10-meter diameter circular plots. Within the plots, I measured again all the mangroves taller than 1 meter, and then deployed the quadrats for mangroves shorter than 1 meter. I conducted the freeze burn by looking at between 0 to 100%. So 100% would be completely freeze burn or damage, as you can see on the mangrove on the left. And then to assess for mortality, or is the mangrove officially dead, I conducted what I called the bark test, where I scraped the roots of mangroves or the bark. If it was green on the inside, the mangrove was still alive. And if it was brown, the mangrove was officially dead from the freeze event. Now, before I get into my data, I just wanted to show some pictures. I know this is what people are mostly interested in. So here is the pre and post winter freeze pictures. This is Piney Island, my northernmost site. You can see here on the left, taken on December 13th, and then almost a month after the freeze event on the right. Here is Dog Island. So here's a picture of a black mangrove, again taken on December 13th. And then almost a month after, you can see a little bit on the interior section of the mangrove, we have some freeze damage. And then here is a black mangrove in my southernmost site of Pilot's Cove, again taken on left December 12th. And then on the right, you can see it's 100% freeze damage. But if you look closely at the bottom, you can still see some green and alive mangrove saplings. And then throughout this, while I was doing the freeze damage and mortality assessments, I did see a lot of variation in the freeze damage. Most importantly, looking like there might be some sort of effect on mangrove height. So you can see here on the left is a tall red mangrove, but surrounding it are shorter mangroves that were still alive. However, there still are some mangroves where I was seeing where they were exactly the same height, less than a foot apart from each other, as you can see here in the middle. The one on the right, 100% freeze damage. The one on the left was 0% or completely fine. And I still don't have any explanations for that. And lastly, I just want to make note that the mortality assessment was conducted almost 1 year after the freeze event due to the resprouting of black mangroves. So black mangroves do have the ability to, once they get freeze damage, they do have the ability to resprout. So you can see here on the left is just some resprouting, and then also on the right, this was taken during the summer growing season, and you can see it's already starting to resprout on the bottom. So now when looking at the data for both freeze damage and mortality, we start seeing this happen with the mangroves in this region starting at negative 4.1 degrees Celsius for both species. And then when looking at just the black mangroves, almost 50% of all the black mangroves that I sampled had some degree of freeze damage, whether that was 1 to 100%. And of that, the average damage for black mangroves was 32%. Versus for the red mangroves, almost 83% of all the red mangroves that I sampled exhibited some degree of freeze damage, and of that damage was almost close to 50%. And lastly, I just want to make note that this will hopefully be one of the first published air temperature data that we have for red mangroves for freeze damage and mortality. So next, I just wanted to show a bit more of the data in regards to freeze damage and mortality. So now just showing my site of Alligator Harbor. This site again only has red mangroves. Almost 90% of all the red mangroves I sampled exhibited some sort of freeze damage. And for mangroves specifically at this site, there was a predicted mortality threshold of 112 centimeters. So you can see here on the graph, we just have mangrove height that I measured, and then the probability of mortality. So if a mangrove was still alive, it was ranked a 0, and if the mangrove was officially dead, it was ranked a 1. So in other words, mangroves at Alligator Harbor that are taller than 112 centimeters have a greater than 50% chance of being officially dead during this freeze event. Very similar for Dog Island, 67% of all the red mangroves that I measured exhibited some degree of freeze damage. And very similar to Alligator Harbor, it had a very similar predicted mortality threshold. So in other words, red mangroves at Dog Island that were taller than 113 centimeters had a greater than 50% chance of being officially dead from this freeze event. And lastly, Pilot's Cove. I do have data for all my sites, but again, because I'm a little bit short on time, I'm just going to be showing a couple of the sites. So at Pilot's Cove, this is my southernmost site. 90% of all the individual red mangroves I sampled exhibited freeze damage, and the mortality threshold was a bit taller at this site at 167 centimeters. Now, this was just for the red mangroves. And I do have data for the 2 sites that have black mangroves. However, I was not able to get a discernible mortality height threshold. So you can see here on the graph, again, we have mangrove height, the probability of mortality. And for Dog Island, it was giving me a mortality threshold of about 175 centimeters. However, I did not measure any mangroves taller than that to be officially dead. Very similar to Pilot's Cove, again, it was giving me a mortality threshold where I didn't even measure any dead mangroves above that height. Most of the death for black mangroves was occurring at about 100 centimeters, so at that middle range height. And this actually has been studied before, where Oslin and others looked at the 3 vegetative life stages from seedling, short, and tall, and the resistance to winter extremes or winter freeze events. And he actually showed that the short life stage mangroves actually were less freeze resistant compared to the tall. So in other words, the taller the black mangrove, the more resistant it became to freeze events, which is what I have been seeing a bit more for my mangroves at our sites. So for the takeaways, going back to my hypothesis 1, the physical structure, it was supported. Even though we saw small variations in the tall mangrove heights, there was a bit greater for the short class, and we saw the most dramatic differences in stem densities for both class sizes. So even though we are seeing the red mangroves grow a bit taller than black, we see the black mangroves dominating in abundances. And again, this could be due to establishment times or differences in growth rates. And the reason why we might be seeing red mangroves grow taller than black mangroves is it actually has been shown before that black mangroves at their range limits tend to grow outward rather than up to outcompete for the salt marsh vegetation. So for hypothesis 2, for nutrients, it was not supported. There really were small differences in the nutrient content or potential limitations that explain the differences that we're seeing in the physical structure of mangroves. And the only significant difference that I was seeing was between my site of Unit 4, which is a picture here, where we have mangroves in the backyard of people's houses, so really close to urbanization, other runoff, compared to Dog Island, which is my most secluded site. So for temperature, hypothesis 3 was definitely supported. And again, just want to make note that we start seeing both freeze damage and mortality when temperatures reach -4.1 degrees Celsius across my sites. And we do see that black mangroves did have a bit higher freeze damage threshold. Black mangroves, again, do have the ability to resprout. And then for red mangroves, what we saw taller than 1 meter were officially dead after the freeze event, and this might be a long-term effect. So for people continuing the study after me, we might see diminished counts of red mangroves due to this. And a reason why we might be seeing that the shorter red mangroves surviving a bit more is that they might have been sheltered by the surrounding salt marsh and mangroves, possibly due to the high tide providing a bit more warmth, as well as studies have shown that mangroves and other vegetation near the soil surface is a bit warmer as compared to getting taller. And then in contrast, black mangroves, it varied a bit more, and this might only be a short-term effect in this region. And when looking at it, so like what is the fate of mangroves in this region, is that repeated freezes might have the potential to set back the encroachment of mangroves, especially in the interior sections. So as I mentioned, we were seeing micro-scale temperature differences compared to the fringe and interior. So if freezes do continue, we might only see mangroves along the fringe end or right at the water's edge, and just showing the importance of micro-scale air temperature differences and how it's playing a role in the distribution of mangroves in this region. And lastly, this research is important to document and understand how mangroves are changing and what will this region look like in the coming decades. Because with mangrove expansion, it is going to bring change. It's going to transform the environment, not only for salt marsh vegetation, but also potentially for oyster reefs, which is what we're already seeing happening in Apalachicola Bay. So with that, I just wanted to say a really big thank you, first and foremost to my advisor, Josh Breithaupt, for the help and support throughout this project, and also to my co-advisor, Thomas Miller. And I also just want to say a big thank you to my lab members who made all this field and lab efforts and helped me out with this. And then also a big thank you to the funding that was supported not only through Florida State, but also through our Coastal and Marine Laboratory. And with that, I can take any questions if we still have time.
Okay, yes, we do. We have plenty of time. Thank you so much, Rachel. That was great, very succinct too. Does anybody have any questions? You can put it in the question box or you can come off mute.
Do you want me to stop sharing my screen or keep it on?
No, that's fine. You can keep sharing your screen. I think that's fine. If you want to raise your hand or put a question in the question box, I'm trying to make sure everybody is off mute. Okay. Give them a minute to think of some questions. Sometimes it takes a while to find the question box and get it in there. And that's why I like just getting everybody off of mute. Having trouble. Don't seem to be able to get everybody off mute. But if you raise your hand, I can see it. Oh, we have a question here from Richard Blake. He says, have there been any measurements since 2025 winter after we had snow? Oh, I think you're on mute. Maybe? I don't know what's happening with this for some reason. Bear with me just a second. Yeah, go ahead. It just put you on mute when I did everybody else.
Okay, can you hear me now?
Yes, thank you.
Okay. So I just want to confirm that the question was, have there been any field measurements from that recent freeze event where we did get snow in the area, correct?
Yeah.
Okay.
Well, we actually didn't get snow. We only got sleet, but it's close enough.
Yeah. No, there hasn't been any measurements. Yeah, I talked with Josh after that freeze event, and he was not able to get a team out there to conduct any freeze damage assessments or mortality. He actually said that he did actually go out to one of the field sites, and the mangroves didn't actually look as bad compared to my freeze event. But yes, unfortunately, no field measurements taken from that freeze event.
Yeah, it still would be pretty relevant. And then Kim Miller said, thank you, great information, very interesting. Next question is from Megan Lamb, and she says, if you did have the opportunity to continue the study long-term, endless time and money, what questions would you be asking about this northern mangrove population long-term?
Yeah, no, that's good. I think if I could continue this, more questions I will look at is definitely deploying more temperature loggers. Maybe looking at temperature loggers that are recording, maybe, for example, every 5 minutes or every minute. And then also looking at maybe other environmental factors, because as mentioned, doesn't look like nutrients really had an effect. So potentially looking at maybe water temperature or salinity, or maybe just a couple different other environmental factors, taking this project a step further for long-term monitoring.
Great. All right. Give it just a minute. Not seeing any other questions, but we'll give everybody just a minute more to see.
And I can always leave my email in the chat box or something if people want to.
That'd be great.
After, of course.
Yeah.
If they have any questions further.
That sounds great. Or you can contact me. I have Rachel's email. All right. Well, I'm not seeing any other questions, and we thank you so much. It was a great talk. You were very well prepared and great slides. Very easy to understand.
Thank you. Thank you again for having me talking this. This was really great.
Yes, yes. And next month we are scheduled for April 24th to have a Sci Cafe. It is hopefully going to be related to the Conservation Corps efforts of creating structures for oysters to attach to, especially related to shoreline restoration. So stay tuned, and we thank everybody for coming.
Yes, thank you again.
Okay, thank you, Rachel.
Thank you.